Hello guys, welcome to Ashutosh Presenter. So hello guys, welcome to Ashutosh Question. Now today we are going to start chapter name is biotechnology and its application. Okay, biotechnology deals with the industrial scale production of biopharmaceuticals and biological using genetically and modified microbes, fungi, plants and animals. Its application include therapeutic and diagnosis and genetically modified crops for the agriculture processed food bio bioremediation, waste treatment and energy production. And the main three critical research area of biotechnology include: first, providing the best catalytic in the form of improved organisms, usually microbe or a pure enzyme. Second, creating optimal condition through the engineering for a catalytic to act. And third, downstream process technique to purify the protein organic compound. Biotechnology and agriculture. In agrochemical is based and organic culture, genetically engineering cropped and based on agriculture. Green revolution somehow succeed in the tripling the food supply, but yet was not enough to food get growing humans population. Increase the yield have partly and been due to the uses of improvement crop varieties and mainly due to the uses of better management practices and the uses of agrochemicals, fertilizer and pesticide. The for the farmers is in the developing worlds, agrochemical are often too expensive and the further it increases in yield, and with the exciting varieties are not possible using conventional breeding. Plant bacteria and fungi and animals whose genes have been altered, manipulated, are called genetically modified organisms, GMO. And GMO are useful in many ways. Genetic modification and they made crops more tolerant to abiotic stresses and cold, drought and salt area, and reduced reliance on a chemical pesticide resistant crops, and the help to reduce the post harvest losses and the increasing efficiency of mineral uses by the plant. This prevent exhaustion for the fertility of soil, enhance the nutritional value of food. Example, vitamin A enriched a bio application of biotechnology in the production in pest and resistant plant. The production of pest resistant can be decreased amount of pesticide used, and Bt toxin is used by the bacterium called the bacillum thuringiensis, and Bt for short. Okay, example, Bt cotton and the Bt cotton rice and the tomato, the potato, soybean, etc. Bt cotton क्या होता है? Bt strains are, देखो, some strains of bacillus and the thuringiensis and the produce protein that kill certain insects such as lid, lepidopteran and the tobacco budworm, any armyworm, and the coleoptera and the beetles and dipterans, flies, mosquitoes. The bt toxic proteins exist in active proteins, pro, pro, protoxins, but once an insect ingests the inactive toxin, it is converted into a active from toxins. Okay, from active into an active forms of toxins due to the alkaline pH of the gut, which solubilize the crystal. The activate toxins bind to the surface of midgut epithelial tissue cells and create pores that causes the cell swelling lysis, the eventually causes death of the insect. The choice of genes depend upon the crop and the targeted pest, as the bt toxins are insect group specific. The rest resistant एंड द प्लांट द नीमोटोड, देखो रेसिस्टेंट प्लांट को जानते हैं। और नीमोटोड, एक नीमोटोड होता है मिलोजिन एंड द इकोजनिया and echinacea infect the root of tobacco and causes great Johnson yield. A novel strategy was adopted to prevent in this reforestation, which was based on the process of RNA interference (RNAi). RNAi placed in all eukaryotic organisms by the cellular defense, and this method involving silence of a a specific mRNA due to the complementary dsRNA molecules and that bind and to prevent translation and of mRNA silencing. The source of complementary RNA could be from an infection viruses from RNA genome or mobile genetic element. Okay, transposomes and that replicate via an RNA intermediate. The introduction of DNA was such that it produces both sense and antisense RNA in the host cell. The two RNAs being complementary to each other forms the double stranded RNA, a dsRNA, and the initiate RNAi, thus the silence and thus specific mRNA of the nematode. The sequence was the parasite could not be survived in a transgenic host and expressing the specific interfering RNA, and the transgenic plant hence got protected from the parasites. Biotechnology application in medicine. The recombinant DNA technological processes have made great impact in a area of healthcare by the enabling mass production of safe and the more effectively therapeutic drugs. Further, recombinant therapy do not induce unwanted immunological response as is common and in case of similar product isolated from non-human sources. About 30 recombinants and the therapeutics have been approved for human use and the world over. The genetically engineered insulin. Earlier, insulin used for the diabetes was excreted from the pancreas, slaughtered, and the cattle pigs. Insulin from the animal source and the through the caused some patient to develop allergy or other type of reaction to the region protein. Insulin consists two short polypeptide chain and chain A and chain B and that the linked together by the disulfide and the bridge as shown in the figure. In mammals, including the humans, insulin is synthesized as a program as a pro-hormones like pro-enzymes. Pro-hormones also need to the processed before the becoming uh, fully mature functional hormones, which contain extra stretch called C-peptide. Okay, उसका C peptide is not present in the mature insulin that is present in the during the maturation into insulin. Gene therapy क्या होता है? Gene therapy is a collection of method of allows correction of gene defense and that diagnoses the child embryo and in a child embryo या child को कोई कोई फर्क नहीं पड़ता है has genetic has genes are inserted into a person as cell tissue and a great disease correction of a genetic defect and evolves diversity of normal gene into the individual or embryo to take the function of compensate for the non-functional gene. The first clinical gene therapy was given in 1990 to a 4 years old girl with adenosine dominance and ADA deficiency. Enzymes is crucial for the immune system to function. The disorder is due to the deletion of the gene of the adenosine dominance. In the some children, ADA deficiency can be used by the bone marrow and the transplantation in another, and it can be treated by the enzymes and the replacement therapy, in which functional ADA is given to the patient by the enzymes. But these appropriate were not completely curative as it is first step toward gene lymphocyte from blood of the patient are grown in a culture outside the body. A functional ADCA and the cDNA using a retriever vector then introduced into the lymphocyte and which are subsequently returned of the plant. Because these cells are not immortal and the patient periodically in confusion but an infusion of the such genetically engineering lymphocyte. However, if the gene isolated or from the marrow cell producing ADCA and ADA is introduced into a cell at early embryonic stage and it can be permanent cure. Molecular diagnosis क्या होता है? Recombinant DNA technology and the polymerase chain reaction PCR and enzymes linked immunosorbent assay यानी कि ELISA are some of the technique that are used to the purpose of early diagnosis. Presence of the pathogens and the bacteria viruses is normally suspected when the pathogens has produced disease symptom. During this time the concentration of pathogen is already very high in body. However, very low concentration of a bacteria or virus at a time when symptoms of disease are not yet visible can be detected by the amplification of their nucleic acid by the PCR. The PCR is now routinely used by the detects AIDS patient. Okay, and पेशेंट इसको डिलीट करो, ये ऑलरेडी पिक आ चुका है इसका। एड्स पेशेंट को डिटेक्ट करने में बहुत काम आता है। इट इज़ बीइंग यूज्ड टू डिटेक्ट म्यूटेशन इन द जीन्स एंड सस्पेक्टेड द कैंसर पेशेंट टू। इट इज़ अ powerful technique to identify many genetic disorders. A single standard DNA, RNA, or tagged with a radioactive molecules probes and is a allowed hybridized and is a complementary DNA is a clone cell and followed by the detection using autobiography. The clone having mutated gene will be not appear and on topography film because the probe will have complementary with the mutated gene. The ELISA is based on the principle of antigen body and the antibody interaction. Infection by the pathogens can be defected by the presence of antigen and protein and the glycoprotein and or by the detecting antibodies synthesized against the pathogen. Transgenic animal, the animal that have their DNA and manipulate the processes and the expresses extra foreign gene are known as transgenic gene animals. Transgenic rats and rabbits and pigs, sheep, cows, and fish have been produced, although the over 95% of all existing transgenic animals are mice. Some of the common reasons for producing these animals are mentioned below. Normal physiological development. The transgenic animals can be specifically designed to allow the study of how the genes are regulated and how they are affected normal function of the body and its development. The example study of complex factor and involved in growth such as insulin like growth factor by the producing gene from the other species that alter the formation of the factor and the studied the biological effect and the result and the formation is obtained about the biological role of the factor in the body. Study of diseases. Many transgenic animals are designed to increase our understanding of how genes contribute to development of disease. These are specially made to serve as models for human disease, so investigation of new treatment for disease is made possible. And today, transgenic models exist for many years. Many human diseases such as cancer, cystic fibrosis, and the rheumatoid, and the arthritis, and the Alzheimer's and the biological factor. The medicine requires to treat and certain humans diseases and can be biological product and but such products are often expensive to make. कल देखेंगे. By the introduction portion of DNA or genes which code for a particular product such as human protein alpha A एंटीट्राइस्पिन समझे एंटीट्राइप्सिन क्या नाम है एंटीट्राइप्सिन यूज टू ट्रीट एम्फीसीमा in 1997 the first transgenic cow Rosie प्रोड्यूस ह्यूमन मिल्क इन रिच मिल्क यानी 2.4 ग्राम पर लीटर। तो दुनिया में सबसे पहले कौन सी गाय थी जो दूध दी थी? उसका नाम है रोजी। क्या नाम था? रोजी नाम याद रहेगा? कौन कौन सी गाय पहली गाय थी जिसने दूध दिया था? रोजी। तो आज ये क्वेश्चन घर जाके सबसे पूछना, ठीक है? ठीक है। The milk contained the human alpha lactalbumin and was nutritionally and a more balanced product for human babies than natural cow. The vaccine safety and the transgenic mice are the being developed for use in testing the safety of vaccines before they are used for humans. And transgenic mice are being used to the test of the safety of the polio vaccine and the successfully find to be reliable. They could replace the uses of monkey to test the safety to vaccines. Pardon, safety of batches of the vaccine. The chemical testing, safety testing. This is known as toxicity or the safety testing. The procedure the procedure in the same is the same as the used for the toxicity of drug. The transgenic animals are made that carry genes and which makes them more sensitive to toxic substance and that can be transgenic animals. They are then exposed to the toxic substance and then effect studies. Last is epithelial tissue. Okay, the manipulation of living organisms by the human race cannot go on a further without regulation, and some epithelial standards are required to evaluate the mortality of all human activity that might help to warm and or harm living organisms. Genetic modification of organisms can have unpredictable result when such organisms are introduced into the ecosystem. Therefore, the Indian government has set up the organization as such as GACE Genetic Engineering Approval Committee, and which will make design regarding the validity of GM research and the safety of introducing GM organic organisms for the public services. Please find out the regulatory agency and for the controlling GM organism and taking decision and regarding the same convenient answer. And the bio, what is biopiracy? Do you know biopiracy क्या होता है? जानता है? बायोपायरेसी क्या होता है? बायोपायरेसी में क्या होता है कि कोई जो लिविंग जो होता है ना उसको पायरेसी में सिक्योर किया जाता है, ठीक है, बट आगे देख मतलब उसको बचाया जाता है, इसे बायोपायरेसी कहते हैं. अब आगे देखो क्या होता है. बायोपायरेसी इज द टर्म यूज टू रिफर टू यूज द बायोपायरेसी said resources by the manipulation and the companies and the organization without the authorized and from the countries and the concerned without the compensatory payment. Most of the industrialization nations are rich in funds financially but poor in biodiversity and traditional knowledge. In contrast, the developing and the undeveloping developed world is rich in biodiversity and traditional knowledge is related to the bioresources, and the traditional knowledge related to bioresources can be exploited to the develop and the modern application and can be also used to save time and effort and expenditure during their commercialization. Okay. But okay, so now this video has finished. So you are a part of journey of neet and you have to subscribe a lot as you can, share, and please try to share more and as you can, because this video has taken a lot of practice by me. So bye, see you next video.